Thank you very much for the opportunity to talk today. I'm going to be talking a little bit about, the title of my talk is the next big thing for agriculture from science, and I'll be talking through some technologies, some new-to-market technologies and science platforms we think will make a difference in the future. But I just wanted to, if people will indulge me, to take a minute to look backwards rather than forwards, and to say we've all already lived through the most profound change in the global food system in human history. So these photographs are to remind me that for several 100,000 years, the global food system was hunter-gatherer. That gave us a population of probably 2 or 3 million. 10,000 years ago, in various parts of the world, we invented agriculture, constrained by renewable cycles, by manual labour, and that gave us a population over 10,000 years of about 1 billion. Arguably, sort of 300 years ago, we started the logistic and scientific revolution that gave us the agriculture we have today, the agro-industrial agriculture. But if you look at the graph on my slide here, which is calorie demand from 1500 to 2050, you'll see that really before about 1950, the level of production is absolutely dwarfed by our level of production since. So in the lifetime of many of the people in this room, we've fundamentally transformed the global food system. And I think that's a real achievement, something to celebrate, because the arrow shows what we need to do in the next 50 years, as Barry and many other people have reminded us today. There's a huge job in front of us, but I think it's worth celebrating what we've already achieved. And just to reinforce that, my understanding is that in the last 25 years, globally, we've had a 50% reduction in malnutrition, and that's something really to celebrate for agriculture. So, part of the driver for this transition has been science and technology. And again, I'm going to look a little bit back before moving forward. My organisation, CSIRO, has its origins in the Advisory Council of Science and Industry that was established by the Commonwealth Government in 1916. This worthy group of gentlemen met for the first time in 1917 to discuss agricultural research. And 100 years later, my organisation, CSIRO, still has a very major footprint in agriculture. So, depending how you count, a quarter or a third of our research activity is directly related to agriculture and food. There's an awful lot changed in that 100 years, though, in terms of the science we do. So these are the 3 nerdy slides about how we conduct our research, that I just wanted to, in a very abstract sense, talk about how science and technology is accelerating the same as many other aspects of progress. So when the scientists in my previous slide did research, they would sit down, think very carefully about a hypothesis, think very carefully about how to go and get the data they needed to prove or disprove that hypothesis, and hopefully make a discovery. With the plethora of technology we have today to produce data, we're in agricultural sciences and many areas of science simply inundated by data. So that whole process has been substantially inverted. We can look at the data that's available, come up with hypotheses and make discoveries much faster. So this is accelerating the scientific discovery process in agricultural science as well as many other areas of science. Now I'm going to talk then about the next 2 stages that go from discovery to delivery, to impact in agriculture, invention. So again, those gentlemen in 1917, the plants, the crops, the animals that they were working on were essentially a black box. So you have to try an intervention, see what happened, and have relatively little understanding of how that occurred. Again, there's this acceleration of science platforms, and don't worry about all the terms here, but they all have omics on the end, really understanding in enormous, unprecedented detail what it is that's going on in the plants and animals that we are manipulating in agriculture. And this again allows this extraordinary acceleration of intervention in the crops and plants and invention of new products coming to market. And I'll be talking about many of those in the next few slides. So an acceleration in the invention process based on unlocking this black box in terms of the components of agricultural systems. And finally, innovation. Invention is nothing without the process of innovation that leads off from it to deploy those new ideas into practice. So this is not a linear approach, hasn't been a linear approach for a long time, but there is an increasingly close and complex dynamic between knowledge institutions like CSIRO and universities, governments investing on behalf of the farming sector, the private industries in terms of SME, startups, but also multinational corporations. So a very complex dynamic for innovation. And this slide is partly a reminder to me to apologise for the fact that many of the technologies I'm about to show you, all of them are developed in some way, shape or form with collaborators, and to apologise for those collaborators who I failed to mention running through these slides. The other thing I wanted to just quickly say about this slide is this articulation actually is stolen from a Dutch exploration of what they call the top sector in agriculture and food sciences. So they've done an enormous amount of work in the last 2 or 3 decades to really shorten the lines between industry, government and knowledge institutions to enormous effect in terms of their extraordinary export culture in agriculture and food systems. And while I think in Australia we have a pretty effective innovation system, there's an awful lot we can learn from our Dutch colleagues. So that's a little bit about the way in which science has changed over the 100 years of my organisation's life. We still stand at the global forefront in terms of discovery part, so we're within the top 10 global institutions in science metrics in agricultural sciences, but are putting increasing emphasis on the invention and innovation parts. So what I'm going to do in the remainder of my talk is really run you through what we think are 4 frontiers, broad areas of science that will start having influence on the production systems many people in the room manage over the next decade and contribute to the challenge we saw in that first slide about producing more and producing products, as we were hearing from the previous speakers, that are targeted to consumer needs. So the first category is applying genetic technologies, a range of genetic technologies, both to changing the products that we're selling, moving from commodities to targeted niche products, and improving yield. So this slide tries to capture this idea that we now understand so much more about the components we're dealing with that we can explore interventions into them to produce functional foods that have particular values in the marketplace. So clearly grains are a very, very major part of the Australian agricultural system. This is capturing some of the interventions we're working through in grain. So we can change the starch composition of grains to increase amylose, to change the way our guts, the behaviour of the food in our guts, large gut fermentation, to lower glycemic index, a whole range of functional changes to grain products, because an enormous amount of our calories come from starch, coming into functional foods that you'll now be seeing marketed to address a lot of the non-communicable diseases, diabetes, high blood pressure, obesity, that are a function of our current food system. So again, minor changes that can have profound impacts on producing new products, differentiated products, but also have health outcomes. So starch and polysaccharides, proteins. So many people, about 1% of the global population are celiacs, can't eat gluten. About 10% of the global population are gluten avoiders. And gluten is a protein in many grains. We can quite easily, well, not easily, we have switched off gluten production in a number of grains, so we can produce genuinely gluten-free barleys and other grains that allows us to develop products. We're very proud of the beer shown up here, which has just been released this year, which is the first genuinely gluten-free beer that meets the German beer purity standards, where beer has to contain, I think, water, barley and hops only. So that opens up a beer market. Germans love beer. 10% of Germans presumably can't drink beer because they're gluten avoiders. This is a huge market. It's a $10 billion market globally in terms of gluten intolerance. The final bit on this slide, micronutrients. So the outside of the grain is where a lot of the micronutrients sit. And again, we can intervene in the size and thickness of these ailerons to change the micronutrient content to meet some of the particular poor nutrition outcomes associated with some grains. So there's a lot of work we're doing in really changing the nature of the product that's coming off our grain farms. And again, in partnership with a number of research and development corporations, lots of companies in commercialising. Now here are some examples domestically of new products through genetic manipulation. Those examples previously were not GM, they're through traditional breeding methods. I'm going to run through these very quickly. Safflower is a very marginal crop in Australia. We've recently worked on using RNAi to silence some genes in terms of the oil that produce the oil content of safflower seeds. So we're getting super high oleic acid oil, 93%. And this is used for a whole range of biomedical, pharmaceutical, electronics applications, and is a very, very high value product. So safflower oil goes from being a product of marginal value to an extraordinary niche product, a very high value product. And this is now being commercialised by a company called Go Resources. We heard earlier today about the fact that the omega-3s in the fish that we eat, which are good for us, are actually accumulated by fish from algae. So we spent some time going through the Australian National Algae Collection at CSIRO, finding the genes that produce omega-3 in those algae and transferring those into canola. So this is a potentially extraordinary intervention into the aquaculture industry where as soon as you start feeding fish with non-algal or feeds that haven't accumulated omega-3s, you start losing that omega-3 content. We can now potentially produce that on land, feed that to fish and increase the sustainability of aquaculture production. So a single hectare of canola produces roughly the same omega-3 as 13,000 fish that would otherwise be going to the salmon farms that you're eating. So again, really potentially quite a transformational change in terms of the sustainability of a very fast-growing sector in aquaculture. Third example here is and that again is under commercialisation development with partners in GRDC and USEED. Third example here is much earlier on. You all know that oils from plants are almost entirely from oil seeds. The seed is where we accumulate the oil. Some of our scientists have been looking at engineering plants to express oils in the vegetative material. So we're getting tobacco plants, just because they're a convenient experimental crop, which have 25% rather than less than 1% of their dry matter as oils. So this is an extraordinary change in the value of those crops, and again, one that may have a whole range of applications in fodders, high protein, high value fodders, and potentially through into a whole range of other biomass, biofuel applications. And again, challenging science, but science that can increasingly rapidly be rolled out. So those were GM technologies, but not going into the food system. I have here, though, two examples of GM technologies that are targeted at human consumption, and these are potentially addressing the issues of food security in Africa we have been hearing before. So the first is looking at locking in hybrid vigour in crops. The photograph here is obviously maize to show the incredible impact of crossbreeding on maize yields. But we're actually going to be looking at doing this in cowpeas in sub-Saharan Africa for reasons I'll talk about in a second. So it turns out that it may be possible to cross two lines of cowpeas to create a variety with this hybrid vigour, but instead of then having the degradation in hybrid vigour as subsistence farmers use the seeds for future generations, actually switch off the sexual crossing that happens in cowpeas to asexual reproduction. So we're locking in hybrid vigour for those crops. Cowpeas for sub-Saharan African farmers, subsistence farmers, are an incredibly important source of protein, and so there's a potential here to dramatically increase yields on what is a key source of protein for many of the people we were hearing before. A second example is also in cowpeas, and this is about transferring the Bt genes that are used in Australian cotton and cotton around the world into cowpeas to reduce the impact of insect pests on them, particularly borers. So again, cowpeas are fundamental for subsistence farmers in sub-Saharan Africa. This technology can increase yields by a factor of 4 to 100-fold, depending on the impact of insects. So challenging science in both those cases, and equally challenging pathways to deployment in terms of regulation, acceptance, and so on and so forth. But opportunities for technologies to quite radically address some of the nutrition challenges in Africa. So I'm going to speed up a little bit. That was applying genetic technologies. Another category is a whole range of novel materials that are coming on stream that may have significant influence in production systems here. So I'm only going to talk about one example, and it's not a very glamorous photograph, but it's a potentially high-impact example, which is these sprayable biodegradable polymers to reduce or radically reduce evaporation from soils. So rather than using plastic sheeting in high-value agriculture, we have the potential to have these sprayable polymers that are biodegradable, not polluting, and to some extent can be controlled. So we can control the degradation process so that these polymers last for 3 or 6 or 12 months until canopies are closed and radically reduce transpiration. So a different technology addressing an age-old issue, particularly in Australian agriculture, about maximising the return per millimetre of rainfall or per megalitre of irrigation. Another frontier, and again I'll have some examples of this, is intervening in a lot of the microbiological sort of ecology of the systems we're dealing with, so bioactive agents to manipulate those biomicrobiological processes. We've heard a little bit today about the use of biofungicides. Clearly there's a significant challenge in a lot of pest and weed control in terms of using chemical fungicides, herbicides, other pesticides is an incredibly complex ecology of microbes, but there are opportunities and increasing opportunities to intervene. So this slide simply shows a healthy canola seed on the left-hand side, canola treated, infected with sclerotinia, and canola infected with sclerotinia, but with a microbial agent added as well to the system. So it's essentially knocked out that infection for the plants. Very difficult to scale into practice, but there are, again, accelerating science that allows this to occur into the future. Again, some other examples, again, looking at microbial activity, in this case in prawns. So we have an aquaculture feed which has been commercialised in recent years, that for a 2% addition to prawn feed can result in up to 300% increase in yield. So this is not about nutrition or calories for the prawns, it's about changing the way the prawn's physiology, their gut physiology, processes the food they receive. So again, an 300% yield increase is an astronomical achievement for a single step in intervention. Today seems to be the day of seaweeds. My third quick example of bioactives is in relation to seaweeds to reduce methane emissions in ruminants. So we've heard how serious a problem methane emissions from livestock are. It's a great discovery, a piece of work a colleague of mine, Rob Kinley, worked with a farmer in Canada who noticed that his cattle that had shore access did better than the cattle that didn't have access to the seafront. Turns out they were eating seaweed. Turns out that seaweed was knocking out the methane genesis in their rumen and turning that methane into meat. The energy was saved. We then screened a whole lot of seaweeds and found one genus in which 1 or 2% addition to seaweed diet, to ruminant diets in intensive feedlots can reduce emissions by possibly the order of 80 or 90%. So again, a very significant step forward by intervening in that microbiological activity. Now, the final frontier I'm going to talk to is digital agriculture, and frankly this is the one I find most challenging. It's not surprising to me that many of the pitches we heard earlier today were using ICT technologies, and this is frankly a disruptive wave that's coming over the top of science as well as agriculture as well as many other sectors. So there's a slide here to look at those 3 blue circles. That's the number of internet-enabled things in 2006 and projected to 2020. So this is this idea of the Internet of Things, and that's fairly abstract. Picture at the top shows a bee with an RFID device on it. That's a member of the Internet of Things as soon as it has that RFID device. And we have a display here on monitoring traps for fruit flies, again, which are based on sensorizing the landscape with Internet-capable devices. This really is an extraordinary addition of information and capability that underpins the potential for digitally enabled decision-making that we're hearing about in those pictures today that, frankly, when I started my career were a pipe dream. But again, there are technologies at each point in this in terms of understanding the resource space, in terms of proximal sensing, satellite technologies, proximal sensing, so measuring crops and animals on a real-time basis to feed back into the type of decision aids we'll increasingly be using. So the breakthrough technology in each of those spaces, and including that map at the top, I'm told potential for step changes in the efficacy of our weather forecasting and seasonal weather forecasting in the next few years. So this means there's a very complex technology innovation ecosystem in terms of developing these technologies, so sensing technologies, all the analytics and informatics, artificial intelligence, machine learning, model data fusion, and the application platforms, which are really accelerating away in a sense that is frankly very challenging for us as well as many others. So huge opportunities, but huge challenges, and this is just a set of some of the companies that are working on some of these components. So a real challenge for, I think, Australia and globally in the next few years to see how we best realise this set of technologies. Now, I've been talking entirely about technologies on farm in terms of pre-farm gate production, really. The other dimension though to emphasise is the opportunity for this digital integration and disintermediation we've been hearing so much about today to have profound impacts along the value chain. And in terms of traceability, in terms of food safety, issues of provenance, in terms of food processing technologies, and signals back to the farm. Again, there's things here we're really thinking about very carefully in terms of where we put our investments in technologies. So I'm 3 seconds away from my time, and this is my last slide. I really just wanted to emphasize that what I've been talking through is under those 4 categories, so genetic technologies, novel material sciences, biological, so bioactive agents, and this whole ICT sensor and data and analytics revolution. The technology we're developing are only of any value where there's that deep partnership with people in industry to understand their pain points, with the people who can take these and scale these, commercialize these technologies and apply them to practice. So thank you very much.